about the incredible growing and shrinking of the human brain in about 40 minutes. And I did this talk with this whole slide that have lasted 2 hours, and there's been some that have lasted 20 minutes. If it's too fast or going too slow, feel free to interrupt me as well. I'll try to be on the time as well, so we actually have time for questions. I'll speak a little bit slow. About the size of the human brain, and this is really a question that sort of sparked a lot of the research that I'm going to be talking about today. And this is really essential. Basic question of how big is the human brain, and it's basically that after 20 or 30 years of research, we can actually have a quantifiable answer to that. There's two pictures like this, perhaps, if you've ever opened the New Scientist or maybe a National Geographic and compare the human brain against brains of other species, and they'll tell you that there's obviously this is a very rough estimate because we can't actually count those. Of this many grams or this many cc's or this many cubic millimeters or whatever. It's a rough estimate, or it's an estimate of that one specific brain that they've looked at in that context. Because there's actually quite a lot of variation. Let's see if I can get to the next slide. There's quite a lot of variation in what the brains actually look like, in terms of their shape, their sizes. There's a lot of variation in terms of between sexes. There's variation obviously with age, and I think that's the thing we're going to be talking about most today. And just to illustrate like how big those changes really are, I have this little clip of, this is basically a fetus growing from about 15 weeks old to essentially birth. And you can see how much, very subtle, it goes very gradually, but you can see like from the sort of first image to the last image, how much the brain actually sort of changes in size and in shape. And that makes it really hard to sort of quantify these different properties. But if you really want to answer the question of how big is the human brain, we have some examples that have sort of served as a reference point for this. And I think one of the perhaps most well-known examples is this idea of a growth chart, which people are probably familiar with, might have seen it in some shape or form. And we have these everywhere for measuring height or weight or even head circumference, essentially. So why don't we have a similar chart for human brain size, or human brain size variation, where we can just say, well, this is roughly the size and this is roughly the variation that we see in a population. Well, the first reason we don't have that is that actually measuring size or any other property in the human brain is quite complicated. So we basically have all these different fancy machines that we can use to measure different properties in the brain versus just a simple tape measure that you effectively use for height or weight even. The second thing is that there's so many things we can actually measure in the brain. So I realize this is a very convoluted slide, and it's just an overview to see like how big this space of things that we can actually measure with brain imaging really is. And for today, I'm mostly going to focus just on sheer volume of the brain, but I'm going to talk a little bit about volumes of different parts of the brain. So I will talk a little bit about volume of just gray matter versus volume of just white matter. And I will talk a little bit about a property called cortical thickness, which is the thickness of the outer layer of the cortex, which is another thing that we can actually quantify with brain imaging. Before we get into all of that, I think it's also good to probably emphasize why it's useful to have a chart like this, or why it'd be useful to have a brain chart. And the first thing I think that is something that I've already kind of shown in that little movie clip is that we see that the brain changes a lot, but it changes a lot, especially during early development. And we haven't really captured how quickly it changes, how much variation there is in that. But what is interesting, we do know that a lot of psychiatric or neurodevelopmental conditions start around that same time where we hypothesize that there are these massive changes in the brain. And there's a lot of theories that this period of sort of rapid development, or these periods where there might be a lot of variation in the population, are also key periods where people might be vulnerable to sort of end up on an atypical trajectory, or might end up on a sort of a risk disease trajectory. And that doesn't only pertain, I would say, to early development. It also pertains to old age, where we can see that maybe at some stage people start deviating from a sort of healthy pattern of aging or maturation. And I think there's several ways that you can think about this. So this is a nice, I think, infographic to illustrate some of the scenarios that you can talk about this in terms of development, but I think to some extent they also pertain to sort of aging and maturation. And that's this idea that there is a sort of a typical, very rough population, typical pattern of brain development, but there's various scenarios where this might go wrong. So this example is specifically in the context of autism, which is a field that I've done most of my research in. But I think as an infographic, it kind of serves for any kind of neurodevelopmental or aging process. And so that's basically this idea that you have these different scenarios. So you can have a sort of a precocious pattern of development where the brain maybe starts sort of growing a lot faster than is typically the case, but then it halts a bit earlier and you end up with a pattern where you actually have a sort of underdeveloped brain. There could be a delayed pattern of development where it simply starts growing a little bit later and then sort of speeds up a little bit, but it also keeps growing a lot longer. There could be a halted pattern of development where at some stage during this sort of maturational pattern, it basically stops developing in a typical manner. There could be an altogether failure to mature. That's not something we see a lot in practice, but it's another possible scenario. And there could be some kind of ectopic pattern of development where it basically just really grows out of control, so to speak. So I would say the last two scenarios is not something you see a lot in practice, but there are obviously like quite extreme cases every so often as well. So before we can actually try and see whether these are the types of scenarios that we see in practice in clinical populations, we obviously first have to understand what the typical pattern even looks like. And to date, we don't really have a full understanding of that. Now, I should highlight that I'm by no means the first person to have thought about this. And I think the idea of having a very standardized, rigorous sort of normative reference chart for the developing of the human brain, it's something that's been on the minds of many. I think the project that I'm going to be talking about has probably been in the works for a good part of 7 years. But I think as long as there's been sort of technologies available to actually measure the human brain in vivo, people have thought about doing some of these things. So here I'm just highlighting a few, I think, mostly examples that have sort of inspired me or have inspired my work, of people really trying to quantify what do these sort of typical trajectories look like. So there's work from Armin Rasnahan in the US, who's done a lot of work trying to see like what are the sort of sex and gender differences in terms of how these brains develop. There's a very influential paper from John Gilmore that I've pictured that I've shown earlier, which is mainly a hypothesis based on sort of stitching together various different research findings and really trying to sort of outline like what are the periods during early development where change happens quickest. And then there's a lot of work from Nico Dozenbach also in the US, who's done a lot of this work, not so much in terms of the volume of the brain, but in terms of different functions or sort of patterns of how the brain might be interconnected, how different regions in the brain might be interconnected. So you can see there's a lot of work has already been done. We have a rough idea of what some of these trajectories might look like. And so at this point, you might question, well, if you want to deploy this to study sort of atypical development, or you want to develop neurodegeneration, we have so many fancy computer algorithms these days. Why don't we just throw that at these data and we can say, well, these are individuals that are a little bit atypical or that might be sort of predisposed for a specific disease trajectory. But actually, it's unfortunately not that simple, especially I would say in psychiatry, which is the field where I work in, but also in lots of other fields. And I think one of the underlying factors in why it's actually quite complicated is this idea that in psychiatry, but I would say in sort of clinical medicine probably more broadly, what we treat or what you see in a clinic is not groups of individuals. It's not groups of individuals, it's individuals specifically. And so very commonly in research practice, what we do to study some of these trajectories or to study some groups of people is we kind of clump them together and we try and answer the question whether there's a sort of on average difference between these two groups. But in clinical practice, that is not something that really works. And the way to think about it is that you can have these different scenarios as to how you can stratify different groups of individuals. And in an ideal scenario, such as the one that's sort of highlighted here in the little box A on the left, you have a group of typical individuals who have no pathology, no psychiatric disease, no problems whatsoever, which is quite rare, but they do exist. And you might have a group of cases that is completely different. So whatever sort of combination of variables or measurements you take, you can basically draw a line between these two groups and say, well, these are my patients and these are not my patients. That would be ideal. That would be very simple. I think we'd be very happy to study situations like that because it's very clear cut where the differences are. And we can very easily understand why those differences might be because they're so clear cut. One thing that we see a lot more in sort of clinical practice is something that's a little bit more similar to scenario B, where actually that group of patients isn't really a group of patients. They're just like little subgroups of patients kind of clumped together, and there's a lot of variation between individuals. What we see, especially in sort of developmental psychiatry, is something that's a lot closer to scenario C, where actually the dividing line between patients and non-patients isn't clear at all. And then also there's actually quite a bit of overlap, but there is a little bit of homogeneity within the group of patients. And the other scenario is scenario D. I would say there's probably a scenario E, but we'll get to that later. There's scenario D where there is a clear group of sort of non-patients, but all the patients are totally unique, and there's nothing really that sort of makes them stand out as a heterogeneous group. And even some of these patients, as you can see in the little blue icon there, might be indistinguishable from individuals that have some kind of diagnosis. And so the ideas kind of emerged that if we want to do science that is relevant in clinical practice, we have to have a method that measures something that is relevant for an individual and not something that's relevant for a group of people. And that's where this idea of normative models kind of comes into play. So it goes back to this idea of a growth chart, because if you think about the idea of a growth chart is you have a standardized sort of population reference, but you can take a single individual, measure them, and you can put them on that growth chart and say, this person is like in a quite high percentile of the population or in a very low percentile of the population. And I would say, especially in sort of a context where you measure someone multiple times, you can actually think about maybe some of these things are an indication that something might be going wrong, where you can see someone deviating from the pattern that they're at. I should also say very much upfront that in the context of looking at brain volumes, having a big or a smaller brain isn't really indicative of something on its own. I think there's a lot of research looking into big brains associated with IQ or other things. We don't really find that there's a lot of evidence to suggest that it predisposes people one way or another. And it's definitely a question that's come up a lot in our work. Like, what does it mean if I have a big brain or a small brain? I would say on its own, it doesn't necessarily mean anything, but it does mean something when we see that it starts deviating. So you can think of this if someone's measured at some point in time and their brain might be in the 20th percentile, nothing wrong with that. They come back 2 years later and suddenly their brain is in the 10th percentile or in the 90th percentile. And that clearly means something has changed, something is going wrong potentially. And so that's this idea of normative modeling, of generating a reference chart and sort of putting people on that reference chart to see like how much do they deviate or where do they sort of fall in the population. Or so again, I could show the same slide again. It's like, well, we have fancy algorithms. We can do this normative modeling. Perfect, right? We can solve this. And now I have a little bit of a convoluted slide. So this is work that we did a couple of years ago that was sort of long in the making. And I realized this is a very convoluted figure as well. What I'm showing here is basically we took a whole bunch of measurements in the human brain. So in this case, we looked at the cortical thickness of a whole bunch of different regions in the human brain. And we used some fancy statistics to project that into a two-dimensional space. So what the picture on the left is showing you, each dot is a person. And it represents sort of a vector of measurements in the brain. And in an ideal scenario, like scenario A that I've sort of outlined in the previous slide, you would be able to just draw a line and say, well, all the people in sort of light blue, those are all the people that have a diagnosis. All the people in red are the people that don't have a diagnosis. And that's basically what you see on the left. That is what a basic machine learning type algorithm or a statistical approach would tell you, that, oh, when we draw a line here, we can stratify these groups based on this combination or sets of measurements. What it looks like in practice is on the right. So this is actually the labels overlaid on the actual data. And we can see that we have a group of people with autism. We have a group of people without. They're quite large groups, about 800 each. So you think there's a lot of variation that we can be capturing with that. But actually, they're quite indistinguishable from each other, and they're largely overlapping. And so we tried in this approach to basically deploy this sort of normative modeling approach. So what I'm showing here is a very basic version of that normative modeling approach, where we first took all a group of individuals that didn't have any kind of diagnosis. We tried to outline for every region in the brain sort of like what their measurements were, and we tried to construct one of these sort of growth chart or normative charts. And then we basically plotted each individual with a diagnosis on top of these charts. And we basically said, this person is this many standard deviations, whereas in this sort of extreme part of the population, and use that as an indication to say they might be a little bit atypical in this sort of part of the brain. And when we aggregate across that data, and we actually, again, sort of compare these individuals against this sort of normative benchmark, what we see is actually a very, very small proportion of individuals with a diagnosis that fall in any kind of extreme form. So across 800 or so individuals that had a diagnosis that we looked at, we only find that there's about 11 or 12, maybe 13, depending on sort of which measure we look at, that fall in that really extreme part of the distribution. What we next did is we kind of took a step back and we said, well, there's been a lot of research, especially in the context of autism, looking to see if they can identify specific brain regions that might be associated with sort of these atypical patterns. And the way that's usually done is, like I said, we're trying to find a sort of dividing line between two groups of individuals. And that looks very much like what you see at the top. So this is a picture of, we had a group of people with autism, a group of people without autism. We measured the cortical thickness in all these sort of different regions in the brain. And then for each region, did a statistical test to see where are the differences. And when we use the full group of individuals, that we get the pattern at the top. We next basically took the step taking our prior analysis and just taking out just those 11 or 12 individuals that showed these really extreme patterns and remove them from the analysis. And we can see that the pattern changes completely. And so to me, this is basically an indication that what the earlier analysis potentially is capturing is just a very extreme but very minor subset of the entire population. And so it might not be all that informative. And this really made us sort of rethink a lot of the types of analysis that we're doing. And it also emphasized the idea that we really need to start focusing on individuals rather than groups. And so there's a couple of challenges that basically came our way from looking at this data and looking at these results. And the first one is that actually in a lot of the data that we had access to at the time, the age range is very limited. So typically, like, people are scanned when they're adults because it's a little bit easier to collect data. People can provide their own consent, which obviously makes things a lot easier if they're taking part in research. But they're generally also, well, we say more compliant. It's easier for them to be very still when they're in one of these brain scanners. But it also created a bias where basically we have a decent understanding of normal brain development, but only in this range of maybe 10 years to 30 or 40 years, because that's the range in which people take part in research studies. The other thing is that we're also estimating all of this data usually from a specific site. Like maybe we have a group of individuals scanned here at Edinburgh's. We might have a group of individuals scanned somewhere in the US. But as I've shown on one of the first slides, there's a lot of variation in these scanners and measurements. And it's really hard to sort of generalize from the data that we have to say, is this really population representative, or are we just capturing individuals in Cambridge and not really people in general? And since I'm not really a clinician, but I fancy myself more of a data scientist, for me, a solution to all of these problems is always just to get more data. And this is really where the Lifespan project, which is the main thing I want to talk about today, sort of originated. So it originated with just a group of us a couple of years ago, basically saying, well, we see all these publications coming out. We see so much data is kind of floating around. There's publicly available data nowadays. Why don't we just go and mine effectively for any data we can find to sort of see if we can get something that is population representative? So I think about 2-3 years ago, we basically started emailing all our friends and colleagues to say, we know you've done a study. Could you give us your data and we'll make this fancy chart out of it? And despite sort of maybe pessimistic views people have about science, we got a very overwhelming positive response. So these are probably just the first 30 people or so that responded to our email, but we're now in a stage where we have basically around 250 different collaborators of people willing to send us data and sort of contribute to this resource. And this is the situation that we're roughly currently in. I always say I've stopped updating this slide because basically we get new data in pretty much every week. I think we're now at a stage where we have brain MRI scans from around 150,000 individuals. And I'm pretty sure we get to 200,000 in the next 6 months or so. I think there's a couple of things to sort of mark, like I don't think you can see the slide on the left, but that's supposed to be a world map, which is kind of, I think, blurred out a little bit. But still, I think the picture is quite clear. If you can imagine a world map underneath that picture, you can probably imagine that most of those dots are data sets that are either from Northern Europe or North America, with a few data sets from East Asia and a few from Australia. So I think that's one of the first sort of objectives in our ongoing research, to really try and see if we can get the diversity of this data set up, because at the moment we're mainly representing sort of Caucasian, North America, Northern European, still mainly people from cities, because those are the people that take part in studies, and that's where those fancy, expensive MRI scanners are based. The other thing to note is that you can see the sort of age distribution in the top left, and we really need people between 30 and 40 apparently to take part in studies, because that's where we're lacking a lot of data. I should also say that that is a little bit of a biased picture, because there's two really big publicly available data sets that we've included in this analysis. One is the UK Biobank, which is a big population cohort in the UK that is aiming to scan around 100,000 people over the next couple of years. And they've already completed about 40,000 of those. So those contribute a big chunk of the data, but they're all around 50 years and older. The majority of them are 50 years and older. And there's a big cohort in the US called the ABCD study, which started scanning people around the age of 9 and is planning to follow them up every 2 to 3 years. But those are sort of those peaks that you see in that plot. The other thing that I think is worth emphasizing about that plot is, if you think about that the earliest slide where I'm showing you that these sort of changes in the brain happen fastest in very early development, because these changes are happening there that fast, and because it's also a period that's relatively difficult to measure, that is probably the period in which we need the most data to actually say something meaningful. Now, it's great that we got this much data, and it's obviously amazing that people were so willing to contribute, but you still have to do something with that. And so we thought we'd start very, very simple. So rather than going straight to all those very complicated measures and all those complicated features that we can measure with imaging, we thought, let's start with a very simple phenotype and try and get a sort of modeling framework in place that allows us to create these trajectories that we could then potentially scale up as we measure other things in the brain. So we started with gross tissue volume. So looking at total gray matter volume in the brain, looking at total white matter volume, and looking at total subcortical volume, so all the subcortical structures combined, and looking at ventricular volume. So this is like the sort of cerebral spinal fluid that is there in the brain. And what you see in the top plot is just the raw data. So we didn't do any fancy modeling. We didn't try to sort of harmonize across all these different scanners. This is just plotting it and basically giving us a feel for what does this data look like and what are sort of the considerations that we might need to put in place in order to be able to actually model these trajectories. And I think the first one, as anyone with a little bit of statistics background could probably see, is that, well, first of all, they're all plotted on a log scale, but despite that, you can also see that there's a lot of sort of non-linearity in this. So it isn't as simple as just drawing a straight line to a bunch of data points to say, well, this is the trajectory. You can see it goes up, it goes down. In some cases it fluctuates. In the case of ventricular volume, you can see that it sort of exponentially increases as we get older. The other thing is that you can also see in this data that the amount of variation changes as we get older. So we can see that when we're very young, obviously volume is quite small, the brain is quite tiny. There's not that much variation in the population. But then around sort of adolescence and especially into adulthood, we can see that there's so much variation in the brain as people get older and older. And so to us, that also meant we need to come up with some kind of way of modeling this data that allows for that variation to change over time. And so that really is like some of these conditions really landed us onto a framework that is called GAML-SS, which incidentally is the framework that the WHO recommends for doing growth chart modeling, but which we found out afterwards. But I'm guessing they probably went through a very similar process of seeing which sort of framework they would use for this. The nice thing about this approach is that it doesn't just allow you to create these trajectories, but it also allows you to have a sort of quantitative assessment of the level of variation in the population and look at other sort of statistically what we call higher order moments of this distribution. And so the first thing you see in the bottom plot is those actual trajectories. So these are the trajectories that come out of this modeling that give you an idea of like how much these properties sort of fluctuate as we get older. And the dotted lines in these plots are very similar to the centile lines that you see in a growth chart plot. Now, as I said, we could also look at some of these higher order moments of this distribution, and that's basically looking at the amount of variation in the population. What are the periods during development and maturation where this variation is highest? And the idea behind it is in those phases or epochs in the lifespan where the variation is highest, we'd expect there to be sort of a higher risk for someone potentially going on towards an atypical trajectory. And we can see that actually for a lot of these properties, that is all very early in life. It's all sort of pre-year 6. So again, these are all on a log scale. The ages are below it, but you can see that a lot of the changes happen essentially pre-year 6. The other thing we could look at is we could look at something we call the rate of growth. So basically trying to identify during which part of the lifespan do we see it growing fastest. And again, here we see that this is very early in life. Perhaps unsurprisingly, I showed you that little movie clip of how fast a fetus grows and how much that actually already sort of resembles a full-grown adult human brain to some extent. So we see that a lot of these, at least for these simple properties, that there is quite a lot of these milestones are in very, very early development. As I said, we try to create this framework with the idea that, well, these are just but 4 very simple properties that we can actually measure in the brain, but there's so many other things we can measure with MRI. Can we use the same sort of approach to also look at some of those other properties? And so that's what we did as well. So this is just showing we can do the same thing, just looking at the total brain volume, which is effectively the summation of those 4 phenotypes or features that I've shown you before. We can also look at properties such as the surface area, so just the sort of size of the outer layer of the brain. and things like cortical thickness, but we can also do it in a sort of regionally dependent manner. So we can predefine specific regions in the brain, measure roughly what the volume is of those regions for each individual, and then create growth charts that are specific to specific regions in the brain. And then what I'm showing in the picture on the right here, you can see there's quite a lot of variability in terms of where the peaks of these developmental trajectories are in the brain. So we can see, for example, that in the sort of frontal part of the brain, or the part of the brain that we tend to associate it with cognition and sort of higher level processing, those are the parts that mature much later than sort of the parts of the brain that we might associate it with basic motor or sensory processing. And there's a lot of variation in that. I wouldn't be surprised if we start looking at other properties such as thickness or surface area or other things that we can measure, is that these patterns will shift. And so we tried to summarize a lot of these findings into a single figure, which has become quite a busy figure. But it's basically just outlining what do these trajectories look like, and then like in the little dots and circles you can see in the top plot, outlining where we find these developmental milestones. So where do we find is the peak volume for this property? Where do we find the sort of fastest pace of change? And you can see that pretty much all of those little dots and triangles, they kind of happen pre-early adolescence, and most of them are actually in very, very early development. And so this again re-emphasizes this idea that we really need to sort of try and capture that very early variation much more comprehensively than we're currently doing. The other thing that I think is interesting about what we've been doing, and like I said, like we always set this out as the idea of creating a sort of a normative reference point that we can then use in clinical medicine and specifically in psychiatry. And we were quite lucky that in the data set that we aggregated, there were actually quite a lot of what we call case control studies where people had collected also data from specific patient groups. And so here I'm outlining just a few of those. I think in the total data set, we have about 110 different clinical labels. For our analysis and everything I'm going to show you next, we decided to just focus on groups where we had at least 500 or more patients. That is basically because then we have enough statistical power and statistical variability to make meaningful inferences on this. But what you can see in the bottom plot is in those sort of little black boxes. It's kind of hard to see, I guess, on the white background. On the little black boxes, what you can see is this is the age range in which people are typically diagnosed with some of these disorders. So at the top, you see Alzheimer's disease, which is typically diagnosed sort of after 60. There's major depressive disorder or bipolar disorder, which tends to sort of be usually diagnosed in people in their 20s, schizophrenia, anxiety disorders, ADHD, and autism spectrum disorders. What you perhaps can't see very clearly, unfortunately, is that there's also a little like dotted blue line around those. And so the dotted blue line are actually the age ranges in which we have data on these populations. And what for me is very encouraging to see is that in most cases, we actually have data that is preceding the age at which people tend to be diagnosed. And so we're not there yet, but it's definitely an ambition to think about, if we have data that early, can we actually see that people start maybe deviating from a typical trajectory of maturation before they are actually diagnosed with that disorder? And I think that would make it very interesting, especially in clinical populations where a verbal assessment or a verbal diagnosis might be very complicated. It'd be great to have some of these technologies that could give early indications, especially in conditions where there's also treatment available, obviously. And then the other thing I'm outlining at the bottom here are some of the other, perhaps more conventional instruments that we have to measure maturation. So things like ultrasound, which happen exclusively prenatally, the standard height and weight growth charts from CDC and WHO. We have other scales, but this is really also a little bit sort of usually preaching from my own work that MRI is a great tool because we can actually use it and measure it, and we have data across the full lifespan, which we don't have for most of these other instruments. So we can have a tool that could say something meaningful about early development, but it at the same time can also give useful information about sort of late maturation and old age. And so we tried to do some very basic analysis just to see, can we actually use these growth charts to see if there are clinical deviations? Maybe this is all just almost literally in our heads, and everyone's actually on a very similar trajectory, and we can't distinguish anything. So what I'm showing in these quite busy graphs, I should admit, is basically the centile scores that we extract based on these normative charts. So for each person with a diagnosis, we know where they are in the population relative to these growth charts that we've developed. And what I'm plotting here is the sort of distributions of those for those same sort of major clinical groups. And the dots give you the sort of median or the close to average value of each group. And I think the take-home message for me, or at least what makes me think that we might be onto something here, hopefully, is that you can see that in the groups with Alzheimer's disease or mild cognitive impairment, they tend to be in quite extreme parts of the population when it comes to like their brain volume on these specific tissue types, which is not surprising. It's exactly what you'd expect, because in a lot of these conditions, these are people that have had Alzheimer's or dementias for years. There might be some tissue loss, so you'd expect them to be in a sort of extreme part of the population, perhaps. What to me was more surprising is because this is all relative to their age peer group, you can actually compare like these volumes or these measurements between someone in old age, but also in neurodevelopment, because it's all relative to their peer group. And what to me is quite striking to see that on some of these measurements, we actually see that individuals with schizophrenia show almost similar levels of deviations to those with Alzheimer's disease. Now, I'd not done much research on schizophrenia before, but to me, this was quite astonishing to see. But I would say at this point, this is mainly sort of a confirmation of something that we've known, something that's been quite obvious in scans that people have looked at in the past. But I think it will be interesting to take this further, because the other thing that is a little bit hidden in this graph is those plots show you the distributions of lots of individuals summarized into sort of one single figure. But that is actually hiding a lot of individual level variation. So I'm not sure if my mouse works. I don't think it does, but you can see, for example, in subcortical volume, if you look at the little box around anxiety, you can see that the distribution sort of seems to have two peaks. So it seems to be that there might be a little subgroup that has relatively sort of high subcortical volume, and there might be a little subgroup that has relatively low cortical volume. But if you average them out, then they kind of are indistinguishable from sort of healthy development or healthy aging. And that's something we also observe in sort of autism and ADHD. And I would say specifically in autism, it's perhaps not surprising. I think there's a lot of research on microcephaly or macrocephaly in autism. But if you actually clump all of those people with a diagnosis together, it kind of averages out and they don't seem to be that distinguishable. So one thing we know is that this is actually hiding a lot of individual level variation. We've tried to make this a little bit more complicated. So the next slide is very convoluted, and I'll try and skip over that a little bit quicker. Basically, because at this point, we're just measuring a single property at a given time, and maybe just measuring one thing is never going to be indicative of anything. So we also tried to come up with a way to basically summarize all those different measurements into one unified measurement. And it's basically this idea that you can think of someone can be like in a specific range in one dimension. So they can be like very high on gray matter volume, but very low on white matter volume. And that kind of puts them in a specific point in this sort of two-dimensional space. But this isn't a theory that you can extrapolate to an n number of dimensions, where you can basically put people and give them a deviation score across multiple measurements. And so we've tried to look at that as a sort of single measurement of their rough deviation. And we can see that a very similar pattern emerges, where individuals with Alzheimer's disease tend to be at the extreme end, sort of across all of these measurements. But individuals with depression tend to be like basically spot on where individuals without any diagnosis are. Another thing we've done is, I think this is also like there's going to be a lot of follow-up work on this specifically, is that we've been very lucky in this cohort to have had genetic information from a number of individuals or from a number of studies. And so we've done what we call a heritability study to see whether some of these properties might be genetically heritable. So whether people are predisposed genetically to be in the top or the low sort of centile range. And we can see that when we use centiles, instead of just looking at the sort of raw volumes that we get from these scans, is that the centiles tend to be a lot more heritable, which does indicate that this phenotype or this feature that we're extrapolating from the data is potentially a lot cleaner, and it's removed from a lot of noise. And so it could be an interesting sort of follow-up measure to look for genetic studies to see whether genetic risks for specific disorders might be related to some of these things that we're measuring here. And there's a lot of elements in this whole project that have kind of emerged that we didn't set out to do, but that we kind of had to tackle or had to deal with sort of in the process, that is also kind of speaking for this methodology. And one of them is one of the elements that started one of my first slides, which is this idea of site variability. So every site, every hospital, every place where we take these scans will have their own system in place. And even if they have two identical scanners in two identical places, there tends to be sort of systematic variation between those two things. And this is really a big challenge if you're trying to aggregate data from hundreds of studies or hundreds of sites. There are some sort of gold standard statistical methods to deal with that, but we wanted to deal with that like basically in our modeling framework by simply trying to see if we could adjust the parameters for each site so they'd actually fit on this normative curve. So here I'm giving one example of this big study that I mentioned earlier in the US, which is called the ABCD study, which is measuring individuals across, I think it's 22 or 23 different sites. And you can see in the top plot, these are just the gray matter and white matter volumetric measurements that are measured at each of these sites on average. And you can see there's a lot of variability across these different sites, which shouldn't really be there because all these individuals are matched to each other. They're all the same age. So there shouldn't be any ideally there shouldn't be much systematic variation. There should be some variability, but it shouldn't be statistically significant, ideally. And what you can see at the bottom is, so GAML-SS is the approach we use to try and sort of clump these data sets together. And at the bottom is a technique which is called combat, which is a very fancy statistical Bayesian approach to also try and harmonize data. And it's kind of emerged as the gold standard. So it's what we use to try and benchmark our approach to. And you can see that in both of these cases, there's still variation across the different sites, but there's no longer any statistical differences. And so this approach of generating these charts and then subsequently sort of benchmarking individuals against that also has the nice sort of added benefit of removing a lot of that site-related variation. Which has also allowed us to actually get to 100,000 scans because there's no single location usually where they have 100,000 scans. The other thing, and this is also something I measured early on, is that it might not mean much on its own if someone is in the top or the bottom percentile of a given distribution. It only becomes really interesting once we start measuring people over time to see if they start deviating from these trajectories. We're also quite lucky to have had about 20,000 or so individuals that had multiple measurements over time. And so we could see if we assess this longitudinal measurement, how stable is it? And we see that on average, people only fluctuate about 5% up or down from these centile curves, which is well within the range of even the WHO growth charts, which I think allow for about 10 to 15% of variation around the curves without it necessarily being indicative of anything. We're also, I would say, lucky. We were very fortunate to have a small subset in our data set where people not only had these longitudinal measurements, but they were actually diagnosed or changed the diagnosis during the course of this longitudinal measurement. So we had a small group of individuals that first time they got an MRI scan, weren't diagnosed with anything. They went through an assessment and they were all deemed to be like on a sort of healthy trajectory. And they came back 2 or 3 years later and they might have gotten a diagnosis of mild cognitive impairment or Alzheimer's disease. As well as a few individuals that were diagnosed with mild cognitive impairment and transitioned to Alzheimer's disease later on. And this is really the test case for this tool to see if, well, do the people that transition in towards more severe neurodegenerative progression, do they also show these deviations from their sort of normative aging? And we can see on most of these measurements that they do. So we see much sort of steeper centile declines, people jumping from effectively being in the 50th percentile to being in the 30th or the 20th percentile. Again, I think this is capturing the type of tissue loss that we already know about. So I don't think this is a particularly shocking revelation, but it does open up the question for a lot of future research to see if those deviations can potentially be predictive. Can we see people going on towards an atypical trajectory before the onset of any kind of symptoms? And that's something that we're currently quite actively investigating. Now, the last slide or last bit of the talk is a little bit more in the realms of trying to give back to the community what they've given us. We've been very lucky to have received a lot of this data. We've processed it. It took millions of compute hours here on the high performance cluster in Cambridge. And in an ideal world, we'd just be able to give all of the data back to the community and try and not have people duplicate the work that we have done. Unfortunately, a lot of the data that we were given was given under specific restrictions. We could only use it for this project. We can't share it with anyone else. And that's quite a restriction, on top of which Cambridge is very lucky to have a really good compute infrastructure to do these types of complicated calculations, but others might not be so lucky. So what we instead try to do is very similar to how growth charts are released to the world, and everyone can use a growth chart, do their own measurement, and put someone on that chart. We wanted to build something that is very similar to that, with the idea that also this data constantly changes. We keep getting new data in, so we keep updating these models, and we keep trying to refine actually what we see in some of these models and some of these features. So what we instead came up with is this idea of building an approach that really estimates for a new data set, how much do I need to adjust all these different parameters that have gone into our modeling framework for this data set to fit within the prior model, and then use those parameters to basically give back centile scores for new data. So we've built this, we've made it available online, and the idea is that people can basically process their own data, put all the variables that they're interested in, and that we've also created growth charts for, put them in a spreadsheet, upload them to a website, and get a set of centile scores back from these. And this is working quite well, and I have hopefully a little animation, see if that's going to work. So this is the website that we built around this, which has a long disclaimer. It also allows, I'm going to stand here a little bit, it also allows people to really sort of interact with the data set that we have. So you can look at some of the properties that we've measured. You can look at some of the different sort of clinical groups that are represented in these data sets. See like what their age ranges are, which studies they're originally derived from. You can check the age range per different sort of diagnostic group. We have a little map of all the studies that are included. So for people that want to get access to the original data, there's little links to like application forms for some of these data sets. Because obviously we can't share it, but at least then they know where they can get them from. And then all the charts we also have in sort of digital format, and we keep updating these as we get more data, but it really allows people to maybe zoom in on specific parts of the lifespan that they might be interested in. Look at different properties that we've measured in the brain. You can look at like all these different sort of regional peaks that are shown in an interactive way. So look at different parts of the brain and see like where does this part of the brain, during which part of the lifespan does this part of the brain mature. We can stratify them by male and female. You can look at variants and other things. We have a little diagnostics plot. So diagnostics in this case means just doing those types of comparisons that I've shown between the different clinical groups. Always takes a little bit to load, even if it's a movie, apparently. So what you see in the bottom plot is a very simplified sort of version of the same analysis, comparing like different clinical cohorts, but also comparing between. So we can compare schizophrenia to autism if you wanted to. And the idea is that people can use this to sort of do maybe pilot analysis, or if they have a hunch that something might be happening, they can use this to explore those potential differences. And in the top, you just see the raw data plotted on top of those curves. But unadjusted for all this site-related variation and all these other modeling parameters. And then here I'm showing you can upload your own little spreadsheet of input variables, go to a little input-output tab. And again, it's probably, even though it's a movie, it takes a little bit of time to load. I can't really speed that up. And you'll see a very similar plot of basically your data plotted on top of those reference curves. And you'll see that they kind of fall outside the range. That's nothing to worry about. That just means they haven't really been adjusted for all these different parameters that go in. But the values in the bottom are where you can start to see, are there any differences in these sort of centile scores in my specific population? And this is all interactive. You can click on all the different buttons. And I also can't stress enough, it's all compliant with sort of data governance. We're not storing anything people are uploading to this. I don't think we even know when people are using it, which I would have liked to know in retrospect, but this makes it a lot safer, and it makes the threshold for people contributing to this a lot better. So we're not storing anything, we're not stealing people's data. We do encourage people to send us their data if they want to do more with it. And we've had a good response so far, which is why I think we'll probably get to 200,000 scans, hopefully in the next 6 months or so. And so that's more or less the end of it. There's a few lessons learned from this whole project that I just like to share, I think, in general. And one is that there's actually a lot of data publicly available. Like when we started this project, I don't think we'd ever have imagined that we would have gotten over 100,000 MRI data sets collected. Also, the fact that people are very happy to share. So like I say, we had this massive network now of collaborators, of people that continue to engage with us and to share data with us. And that's been overwhelming. I would say we have a 99% positive response rate to this. And the cases where people didn't share data was basically because the people that they collected the data might have not been able to actually share data with other groups. The other thing I think is you can learn a lot from these very basic features. So we can do a little bit of sort of case control stratification in this. Hopefully you've also seen there's a lot of room to expand. I've given a few sort of tasters of things that we're analyzing, but I think if you think about that slide where we see these case control differences, each little asterisk that's indicating a significant difference is a potential follow-up project to see like what is the individual level variation that is actually causing these differences. There's also a few buts or ifs, I think, in this project. This definitely requires community effort, so we would not have been able to do this without that collaboration. Unfortunately, the data is still very restricted to a specific population, and that's something we're very keen to change. There are a few very exciting initiatives where projects have been funded to bring scanners to remote locations and to essentially geographic locations that are not normally incorporated in studies. And I think that particularly is a very exciting field or a movement to be part of. And then, as I said, we're very lucky in Cambridge to have a very good high-performance computing system that really allows us to do this. And just to illustrate, I think these are all outdated as well. I think we're now sitting on slightly more than 1 petabyte of data in MRI scans, and we've definitely used more than 2 million CPU hours just to process those scans. So it's fortunate to be here, and it's fortunate to have like this network and then these people contributing to that. But that's really essential. And I think the only way to keep growing this is to find ways to sort of give that back to the community and have other people also analyze this data and do things with this. So with that, there's a lot of people to thank. Of course, funders, the people in Cambridge and the sort of close collaborators on this project, but also all the people that have contributed data to this whole process. There is a paper that came out in April this year that has all these people on it. If you want to read all the technical details, it has about, I think, 200. We ended up with more than 200 pages of supplementary materials just to explain these, what I think are hopefully somewhat basic processes. But yeah, there's a lot of detail there. People want to read more about it. I'll leave it now for questions. People have anything. There's a question here. You need a microphone. I'm not sure if my microphone is still working.
Hi, thanks for the very interesting talk. I have a very specific question about the data. How can you tell that the data match the same person, given that you got data from different sources?
I should talk in the microphone. That's a really good question, actually. There's definitely potential that there's some overlap. It depends a little bit on the system. So in the US, it's quite nice that people are often assigned like a national ID when they go in the scanner. And so we know that some individuals might have taken part in different studies and we can exclude them. I think we have one specific occasion where we couldn't be sure and we basically left the whole study out. So there's a big sort of Alzheimer's disease consortium like across the world that also aggregates scans that we were given that had about 12,000 scans from us from multiple sites. And then a few months later, we came across another Alzheimer's data set also of around 12,000 scans. And to us, that was a little bit suspicious. So we didn't include the second one because we weren't sure. And we've since had a lot of conversations with all the different sites that have collected data. And they're basically ensuring us, oh no, we recruited from a different pool in different locations. So it shouldn't be overlapping. But yeah, there is always a potential that people have taken part in multiple studies. I'd say in general, most of the studies that we incorporated were from very different locations. So we have a data set from Cambridge and we have a data set from Manchester. It doesn't seem all that likely that someone who took part in Manchester also took part in a study in Cambridge. On top of which, I think a lot of these studies come from case control studies where people have been recruited specifically to look at autism or specifically look at differences in depression. And there are experiences that people that sign up to volunteer for an autism study probably wouldn't sign up to volunteer for a depression study. That being said, there is a possibility that some people are represented in multiple studies, but I think the chances of that are quite low, and the percentage of overlap is probably quite low so far as that's the case. Any other questions? There's a question in the back there. I should also ask if there are any online questions.
My question is about autism in particular. You said it's very difficult to study young children, for example. So autism manifests itself in young children more than other age groups. So how can you get your data about autism in young children if it's difficult to study them?
I mean, that's an excellent question. I think there's several strategies. So I think one of the projects that we're currently running in Cambridge is a project where we're basically recruiting people that have an older sibling that has already been diagnosed and asking them if they want to take part in research. So that means they're often recruited prior to any diagnosis or prior to any onset. There's a few projects where we know there's a high genetic risks from parents that might have been diagnosed that are recruited into these studies. There's obviously a lot of engagement, I would say, more broadly with the autism community. So with autism charities, with autism stakeholders, trying to reach out and see if they want to take part in research. I would say the challenge very often, and actually on the sort of data collection side of things, is that if you have children with very sort of severe behavioral problems, they're not going to lie still in a scanner for half an hour and not move while we take these images. So I think in a lot of the brain scanning studies that we do, there tends to be an overrepresentation of individuals that are, I would say, on the higher, sort of better off end of the autism spectrum, perhaps. So people that are relatively well functioning that can actually lie still for half an hour. And I would say that's a challenge. One of the things we are trying to do, at least to remedy that, is to also improve the technology itself, where we can get these scans in 2 minutes rather than in 20 minutes, just to make the threshold a little bit lower. The other thing, I'd say this is quite an exciting development. That's something that's only been really possible in the last year or so, is that before I showed you some of these pictures of these big MRI scanners that are somewhere in a big hospital room that take a lot of bells and whistles to get someone in. Increasingly now we have access to small mobile scanners, which are basically like a sort of big cart that you can actually drive up to someone that can take these scans quite quickly, and it's a little bit more feasible and the restrictions are a little bit less severe. And you can potentially do it in an environment where these kids are much more comfortable rather than a big sort of clinical setting that might make them more anxious. So yeah, there's definitely still a lot of challenges, but I think we're getting better and better at trying to overcome some of them, at least.
I ask, your sort of exponential increase in ventricular volume in the aged, is that caused by the brain shrinking or disappearing, basically?
It's partially caused by the brain shrinking, for sure, because it's basically it's a culmination of all the other tissue loss, and that space has to be filled up somehow. People have seen that graph, and everyone's been like, "Oh, well, I'm done for, I'm on that exponential trajectory, and I'm going down." I think the slide is a little bit misleading because it's on a logarithmic scale. So that little spike that happens at the end is basically capturing the last 40 years of life. So it is a lot more gradual than that graph makes it look out to be. I should also say we haven't really found any evidence that it has any sort of clinical impact. So I think it's just a product of healthy aging, effectively, largely. But yeah, I definitely didn't mean to alarm anyone by showing those graphs. I think it might also, in that context, we were like, one of the comments that we've gotten a lot when this paper came out and it got a bit of publicity is people saying like, "Oh, I'm past that peak, it's all downhill from here. I'm a kind of a lost case." But we also find very little evidence that it's associated with sort of your cognitive abilities or IQ or anything like that. So yes, the brain grows and it shrinks, and it does maybe at the sort of boundaries of it have an impact on clinical progression or even like sort of cognitively healthy aging. But as a whole, the fact that you're on that curve doesn't really indicate anything specifically. Any other questions?
The last question.
Okay.
Just given the erratic nature of diagnostics at the moment, particularly for ASD and ADHD and stuff, where some diagnosticians are using 20 years out of date knowledge and some people are up to date, how much is that going to skew the data at the moment? Or you think it's likely to skew the data at the moment?
That's an excellent question. It's something we've had to deal with in a lot of other contexts as well, because we're obviously aggregating data across the world, and how someone's diagnosed in China might not be the same protocol to how someone's diagnosed in the US or in the UK. So I think that's an excellent question. We've, at least for this analysis, we've just gone with the sort of static labels as they were given. I think the important thing to say is that the trajectories that we're generating are not generated from people with a diagnosis. So the trajectories themselves are only generated from people without any kind of diagnosis. And then we estimate sort of the positions of people with any clinical diagnosis on that trajectory. That's basically to make it a population representative trajectory that isn't skewed by one population or the other. So it's exactly to try and potentially prevent that from happening. And then on the second point of that is, because we're then estimating all these scores at an individual level, there's basically always a way to go back and say, well, we can actually use the centile scores, but only look at them in individuals that have been diagnosed in this specific clinic, and they'd at least be harmonized. So I think there are ways around that. I think it definitely impacts those case control comparisons, the graph that I've shown you where we're kind of clumping everyone together. there's a lot of individual variability in there that we're probably not capturing because there is a lot of diagnostic variability as well. But at least by sort of harmonizing, I hope at least that by harmonizing and standardizing the sort of brain measurements that we're taking, it at least gives us a more robust place to start from when we try and sort of make associations with elements of diagnosis. I hope that answered your question.
It's just the other way around. On the controls, you may well have quite a large, if people are interested in getting involved in these studies and they are saying they're not autistic, how do you know that they are actually do belong to the control group and they're actually,
that's an excellent question. So for some of the data sets, we have follow-up information in terms of any kind of diagnosis. People, especially in, for example, the UK Biobank data set that we've used quite extensively in here, that's linked to patients' health records. So we can go back and we can say, well, maybe they come back 2 years later or a year later and they've actually gotten a diagnosis now, in which case that will exclude them from the estimation. I think you're absolutely right, is that there's still a risk in that. And I think it was hidden a little bit, but in the slide that I showed you where you see that individuals transition in a diagnosis in their longitudinal measurements. One thing that I showed but didn't emphasize is that we also plotted all the individuals that were healthy controls at the start and kind of never actually got a diagnosis of any kind. And we can see that they also dip slightly below what would be expected that they were completely sort of typical individuals. So my explanation for that is that there's probably a bit of, like you say, a bit of recruitment bias. These are people that are interested in Alzheimer's disease or in mild cognitive impairment that have taken part in these studies, maybe because they have a family member or because they have reason to suspect that they might themselves be sort of like heading towards something like that. So yeah, I think there's always a risk. We try to exclude it as best as we can. And we try and retroactively go back through the data if there's follow-up to see if something's changed. But yeah, there's always a possibility. At least at the time that they were scanned, they didn't receive a diagnosis. And that's, I think, all we can go with, unless we want to go back to 200,000 people and re-diagnose all of them. It'd be great if someone wanted to pay me to do that, but I don't think that's a very feasible project. I think then we're at the end of it. Thank you all.